Alright, so today we're going to finish our first round of the tour of the cell, and then we're actually going to skip ahead to round 3 of the tour of the cell, because that's a shorter one, and we'll come back to the tour of the cell part 2. So let's start by reviewing a little bit, and then we'll get into some of the newer stuff that we weren't able to get to yesterday. So remember yesterday we talked about the parts of the different types of cells. We talked about that there's prokaryotes and there's eukaryotes. Remember the prokaryotes don't have any organized organelles in them, including a nucleus. And so they're a much simpler type of cell, and as a result of that, they're typically much smaller than other types of cells. And we saw that yesterday with the videos we watched on the cells alive, and we looked at all the pictures that had all the different little things that were on the head of the pin. All right, eukaryotes have many organelles that make them up, and that's what we started talking about yesterday. All right, we also talked about the purpose behind organelles. We looked at the fact that they provide specialized functions to help with certain things. The example we talked about was how some of the cells in organisms have cilia or flagella that help to be able to either move the organism itself or to be able to move things across the outside of the cell to be able to help transport it from one location to another. Remember we talked about the digestive system and some of the things that you find in the digestive system that make it so that happens. We talked about it provides containers within the cell so that you can have different jobs going on at the same time that require different environments. For example, the chances are this one of you texted me a question about your formal lab. To go with that, you're going to be looking at something like the lysosome. We talked about the fact that the lysosome needs its own separate pH so that it can have the digestive enzymes that it needs to be able to break down things that are inside the cell. So we need these organelles. They provide containers for separate environments, and it allows then for additional membranes to be present so that chemical reactions can happen. Prior to this video, you watched the one with Mr. Boozman that he went through, and he talked about why the cell membrane helps to increase the surface area to volume ratio and how that's beneficial to cells. And so hopefully at this point that the membranes acting as sites for chemical reactions is a little bit more relevant, because you see now that you have an increased surface area due to all these membranes that are found throughout the cell. That means you have increased areas where some of these chemical reactions can actually be happening. And you have increased areas where you can be transporting nutrients and waste into or out of the cell. All right, and we talked about cells have 3 primary jobs. One of those is for making proteins, one of the jobs is for making energy, and then job for making more cells. And so we started yesterday by looking at what are some of the organelles that help them be able to make proteins. And we got through the general overview of it, and then we're going to get a little bit more specific into some of those organelles today. And then we'll skip the making energy organelles. We'll do that tomorrow. And then we'll talk about how cells make more cells. That's only 2 or 3 slides. It's not going to take us very long at the end of this video. So then we saw the fact that proteins, we talked about, remember, they're the things that do everything inside the cell. All the work that happens to get done inside the cell is done by proteins. We've talked about the fact that DNA, you always hear this kind of gets the glamour and oh, DNA does everything. No, DNA really doesn't do anything other than hold instructions. We've been likening it to a CEO. Now, I don't know if there's any truth to the matter or not, but you hear about CEOs, they really don't do anything. They just are the person that has the majority of the information or has that on file somewhere that's able to detail what happens in that company. And the actual work is getting done by all the people that are working in the factories that happen to be run by that particular business. All right, so the DNA has all the instructions on how to make the proteins. The proteins then are able to do the work for the cells, and then the cells do the work for the creepy little penguin guy with the creepy little sound. Remember, we talked about yesterday, proteins do all the work. All right, so when we look at the cell functions, the big one I want to concentrate on once again today is being able to build proteins. In order to do this, you need to be able to have something that can take the DNA instructions and read them and do something with them. It then take that information to the areas where the proteins are actually going to get made. You then need to have the proteins get folded and modified. We already talked about protein structure and the 4 levels of structure. That's a result of things that happen inside the cell that help to provide the environment for all those interactions between your secondary, or that result in the secondary and tertiary structures of the proteins. All right, then you also have to have it modified. We said it's very similar yesterday to The card comes off the assembly line. The card is ready to be used, but it's not in the shape or in how do I want to say it? It's not been put together in a way that we want to use it yet. You know, we take the car, we send it to the body shop so that it can get painted, so they can get all beautified. The interior stuff gets done. When it first comes off the assembly line and is ready to go, it doesn't have all those things done. Same thing with your proteins. When it comes off the assembly line, they are ready to be used, but there's other things we can do to make it so it can be used better. We're going to talk about some of the organelles that help to be able to make that happen. And then you need to have ways to be able to transport those proteins to either other locations within the cell, or a lot of these proteins then get sent outside of the cell, and they do their work outside of the cell from which they were made in. Alright, so when we talk about doing this, the organelles that are involved in this, we talked about first you have the nucleus, because that's where the DNA is stored. Information from the nucleus is then transferred to the ribosomes. Then that information is used to be able to make the proteins. Once the proteins are made, they get sent into the endoplasmic reticulum, where they're going to get modified, and then they're going to get sent to the Golgi apparatus, where they're going to get sorted and set up for being able to be transported outside of the cell, and they then get packed into vesicles that will then take those proteins to the locations where they are needed. And you can see the graphic of that up above here as well. So we started yesterday, the only organelle we really got through was the nucleus. We said that the nucleus helps provide protection for the DNA because of the nuclear envelope. The nuclear envelope is a specialized membrane that is inside the cell that surrounds the nucleus. Primarily, it's surrounding the DNA to make it so that the DNA can't get damaged by other things. If that DNA happens to get inside of your cell, so if it gets into the cytosol and mixed into the cytoplasm, some of the problems that you can have is your cell's not going to be able to recognize that that is your own DNA, and it's going to see that and it's going to say, hey, you know what, I don't know what that is. I need to get rid of it, because there's certain proteins that's their job. Because remember, they do everything. There's proteins out there, their job is to get rid of stuff that's not supposed to be there. So if it sees that DNA out there, it doesn't recognize it. It doesn't know it's its own. It will start to break it down. That's a bad thing. You lose that DNA, you lose the ability of the cell to do its function. Same thing, you have other invaders that get into your cells by tricking your proteins into thinking they're something that they're actually not. The nuclear membrane provides an extra layer of protection to keep those viruses that get in there, to keep the bacteria that infect some of our cells, that cause some of our infections that we get. It prevents them from being able to damage the DNA as well. When we talk about there's little holes in the nuclear membrane that makes it so that things can be able to pass in and out of the nucleus. One of those things that we really need to be able to come in and out of the nucleus is the messenger RNA. Because remember, the whole process of making these proteins, you start by making messenger RNA, and then the messenger RNA is sent outside of the nucleus where it attaches to a ribosome. I assume that this at least sounds vaguely familiar because we spent a lot of time talking about transcription and translation back in general biology. Alright, so you have the messenger RNA gets made, then gets sent outside of the nucleus, gets sent to the ribosome, where it can now be used to be able to help in the assembly of a particular protein. We then looked at some of the artist renderings. We looked at some electron pictographs and various pictures as well to see the structure of the nuclear membrane. Alright, so I believe this is where we left off yesterday. Also inside of the nucleus, you're going to find a structure called the nucleolus. The nucleolus is the location inside the cell where ribosomes actually get made. Alright, these ribosomes are made up of a mixture of various proteins and something we call rRNA, ribosomal RNA. Back in general biology, we just said that's kind of the glue that holds things together. But really, the ribosome is made up of this rRNA stuff. Alright, once the subunits get made, you can't have it be one big unit because then it's not going to be able to fit through those pores. So you have two separate parts of the ribosome that get made. They then can pass through the nuclear pore and they can go out to the cytoplasm where they can either be found just floating around out the cytoplasm, or you'll find them attached to other things, primarily the endoplasmic reticulum. Alright, so now that we looked at the nucleolus, and one other thing I want to point out, when we looked at the cells underneath the microscope back in general biology, we looked at the onion root cells, and we looked at, we spent time looking at the different phases or different parts of the cell cycle. Well, when you look at the little structure right here, that's the nucleolus. We can clearly see this on some of the slides that we use in class. So when we get the slides out and we start looking at things under the microscope, our microscopes are good enough to be able to see the nucleolus in many of the cells. And often you're going to be able to see it because it's going to be a little bit darker structure inside the nucleus of the cell. So then let's look at what ribosomes do. Ribosomes, their job is they're the protein producers. You know, a lot of people will relate the ribosome to being the factory where things are actually getting made. Now you have this great office building where you have all these files and things, but nothing really happens there. The real work happens inside the factory. While that real work is the ribosome. The ribosome is the factory that's actually making the things that can then be used by the cells to be able to do the work, namely proteins. We already saw there's two parts to the ribosome. There's the RNA, and there's various proteins that work together to be able to make the ribosome to be able to do the job that it's supposed to do. And when you'll find these two parts that you see right here, you'll find those typically not attached to one another. But once the messenger RNA comes in, with that message, it sends a signal that says, okay, we need the two parts of the ribosome to come together so that we can start to assemble the amino acids so that we can then make those peptide chains that are required to be able to make the proteins. Often you'll find these ribosomes attached to the rough endoplasmic reticulum. So you can see that here you have the ER, which looks like just a bunch of folded membranes. And you can see there's little ribosomes embedded in them. You can also find ER that doesn't have that. We call that the smooth ER. Once again, we talk about that in general biology. When you look at the electron pictograph here, you see little dark outlines along the ER. That's evidence of there being ribosomes there. Here you don't see the dark outlines on the outsides of it. That's evidence of it being smooth endoplasmic reticulum. And make sure you notice how small of a viewing area we're actually looking at. 0.08 micrometers. That's quite small. Alright, so the ribosomes, you find two types. You find the free ribosomes. These are suspended in the cytosol. That may be a new term for you guys. The cytosol is the structural area that you find inside of the cytoplasm. Some people will tell you it's the same thing as the cytoplasm. Not quite. We can discuss that a little bit further when I come back. I don't want to spend time on it right now. But anyways, these free ribosomes, you can have the proteins getting made in that area. You also will find them often attached to the endoplasmic reticulum, and there's actually an advantage to that because the next thing that has to happen to the protein is it has to go through the ER to be able to modify it to make it a protein that's functional. So having the ribosomes actually attached to the ER makes it so that you have less distance that has to be covered to be able to bring that protein that's now been made to the area where it's going to be modified to be able to make it even better. All right, and we'll talk about a little more with the ER here in just a minute. In fact, that minute's here. So the ER, moving into that, this is the area where you get proteins that are processed. Here's the other thing though. This is also where your membranes get made. So you have the proteins, they're already made, now we need to process them and make them better. Alright, think of a pencil. You have a pencil, it's already made, it can do its job, but to make the pencil be able to work better, you have to be able to modify it by sharpening that tip so that you can get the graphite exposed so you then can go through and write on your papers. Well, the ER is like the pencil sharpener. It's going to take what's already there, it's going to modify it and make it better so that it can do its job more efficiently. But at the same time, the ER is also responsible for making those lipids, making those phospholipids that are then used for assembling the cell membranes and the various membranes that you find throughout the cell. Alright, so that means you not only have synthesis of things happening here, you also have some hydrolysis going on here because part of what we just saw using the pencil analogy, to make the pencil work better, you have to remove pieces of the pencil. Well, sometimes to make a protein work better, you have to remove some of the amino acids. So we have both synthesis and hydrolysis going on in this structure. When you look at the ER, you can see it's a series of interfolded, essentially membranes. We already saw in the last video, this helps to increase the surface area so you can have more chemical reactions going on at the same time in that little itty-bitty cell. Alright, and so you're going to typically find the ER is going to stretch throughout the majority of the cell. So once again, looking at the pictograph here, you can see the dark outlines as evidence of ribosomes being present. Over in the smooth ER, you don't see those dark outlines. I think this picture shows it better than the last one. An artist rendition of it shown here, and then another pictograph. You see this one's even smaller. You know, it's 200 nanometers. So we're talking 200 times 10 to the negative 9. So we're talking pretty small structures. Alright, the smooth ER, this is where membranes are going to get made. So we said the ER is for modifying proteins and for making membranes. Well, the smooth ER is the location where membranes are going to get made. You have synthesis going on to be able to make the lipids, so you have some dehydration synthesis to be able to make the oils, the phospholipids, the steroids, hormones. Some types of hormones are examples of lipids as well. So those are some of the examples of things that are going to be made in the ER. And then you also have hydrolysis going on inside the ER to be able to remove things from the cells that you don't want there. Examples of that you can see down below. It helps to take glycogen and turn it into glucose inside the liver. It also helps to take drugs and poisons out of your body. The best example of the smooth ER doing this is when people are drinking alcohol and the alcohol needs metabolized. You send it to the liver. The liver has a lot of smooth ER in those cells so that it can more readily go through and hydrolyze or have hydrolysis happen to make it so that that alcohol, which is a sugar, can be broken down into simple sugars so that it's no longer traveling through the system as the alcohol molecule that it came in as. All right, so to continue with this, when you're making new membranes, you can see the process here. I think this picture does a pretty good job of showing that. We have the ER here. You're making these components that are going to help in putting together the cell membrane. They then essentially pinch off from the ER and will then get sent to eventually out to the cell membrane and can attach to the cell membrane. And this is going to happen to be able to help replace areas in the cell membrane that are becoming weak. And it can also, if a hole develops due to some sort of injury, same thing, you can use this to help repair that cell membrane. And so you'll see that the endoplasmic reticulum, it talks about it has the membrane expand and then pieces of it break off. And then those pieces get sent to the cell membrane. Alright, the rough ER, the only difference here is now that you have the ribosomes attached to that means that's going to be more involved in the modifying of the proteins. Alright, some examples of proteins that are going to get made in here. Some of the proteins that are typically secreted or released from cells, like your digestive enzymes, are going to be made in this process. Alright, it gets sent from the rough ER, it'll get sent to the Golgi. Things will then happen in the Golgi to be able to make it so that it can be transported to elsewhere inside the cell. Alright, so the rough ER helps to be able to primarily make protein-secreting cells, and then these cells can be transported, made into vesicles that can then be used for transporting it to other locations. Alright, so Mr. Penguin guy is asking what types of cells are going to have a lot of ER. Well, ones that are going to secrete a lot of proteins. Remember, enzymes are examples of proteins. So if we sit there and we think about what are some cells that are going to have a lot of enzymes in them, mainly the cells that line your digestive tract, because you need those enzymes to be able to break all those foods that we're bringing into our body down into their monomers that our cells can turn around and use for being able to build structures within them. So this is just another example showing that. And this one I think does a good job of showing the advantage to having ribosomes actually on the rough ER. So you have the ribosome here. It's making the polypeptide chains. So these are all links of amino acids. When those start to get made and it gets big enough, it'll actually start to push them into the ER while it's still getting made. And so by having all these folds, you have a lot more surface area for more ribosomes to be able to attach to it. So you can have more of these entering into the ER at the same time, which is going to increase the rate at which you can produce these proteins. Alright, and then you can see there's a couple little vocab terms of this. I guess you go back to it. The little space where these are going to enter into, the space in between the folds on the inside of the ER, we call it the cisternal space. So in some of these diagrams, you're going to see cis. That's referring to the space inside of inside of the membrane, between the folds in the membrane. So then, now that we have these proteins, they've been modified a little bit to make them work a little bit better. Now we have the Golgi apparatus do its thing. The Golgi apparatus, you can liken it, you can see, to the UPS guy, or the UPS guy. I guess I should say gal too, sorry ladies. But the UPS guy, they take the stuff that's already been made, they package it up a little bit more, and they send it off to where it needs to go. They try to protect it to make sure nothing bad happens to that package when you send it off to other locations. That's exactly what your Golgi body's doing. Right? It takes those proteins, it finishes it, sorts it, puts it where it needs to go. You know, you send your stuff in the mail, goes to the post office, all the mail from Sparta goes into the same post office, then they got to sort it, they tag where it's going to go next, they ship it off to where it needs to go. You know, same thing's happening in an UPS location. I know it's UPS, it's just more fun to say UPS. Alright, so then the UPS person or the post office is going to take it and put it into vesicles. Or in this case, the Golgi is going to put it into vesicles, which are nothing more than a membrane-bound sac that has whatever it is it's trying to transport on the inside of it. So you can see here the Golgi, it takes these things that it's trying to package to be able to send elsewhere. It basically wraps it in a membrane by pinching off some of the Golgi with it. And now inside of this structure, you'll find proteins that are going to be transported to other locations, either inside the cell or in many cases sent outside the cell so that they can do their jobs. Alright, so what would be an example of a cell that's going to have a lot of Golgi, where you're going to have a lot of proteins being kicked out to be able to use elsewhere? I think you're going to find a lot of these in the digestive tract as well. Alright, so when we look at the Golgi under the microscope, you can see many folded layers here, very similar to the smooth endoplasmic reticulum. Alright, here's that cyst coming back. It's showing the receiving side of the Golgi, whereas the trans side is the shipping side. So the proteins are going to come in through here. They then go through these series of membranes. They get modified, packaged, sorted, all that stuff. Then on this side is where they get wrapped into a membrane so they can then be sent elsewhere in the cell. Alright, so when you look at this in a more of a close-up view, here's the Golgi body. Proteins come in, they got modified, they got sorted. These are the ones that we want to send to somewhere else. So we wrap them then inside of part of the membrane. It buds off, and now it's getting sent to somewhere else. And this one happens to show it being sent to another location inside the Golgi. Alright, so to wrap all this up, when we're making proteins, you have the nucleus. Inside the nucleus you find your DNA. We then make messenger RNA off from the DNA. So the DNA is the instructions. The RNA comes in and it says, okay, I need to make a copy of those instructions so that I can bring these to another location. So it's kind of like the mail carrier. You give the instructions to the mail carrier, they bring it to somebody else. You take the message, write it all out, send it to the mailman or mailwoman takes it to wherever it needs to go next. So we have the RNA, delivers the message out to the ribosomes. In this case, we can see the ribosomes are embedded in the ER. Goes through the ribosomes. You can see the messenger RNA going through the two parts of the ribosome here. We now have amino acids being put together, because remember, proteins are polymers of amino acids. So now we're constructing our protein. It sends it through the ER so that it can be made better. Remember the ER we were likening to the pencil sharpener. You already have this pencil. Looks fine. It can do its job. You want it to do its job better, so you shave off a little piece of it. Alright. Then it gets put into a vesicle so that it can be transferred elsewhere. That elsewhere is typically the Golgi. And in the Golgi, it'll get modified a little bit more. It'll get repackaged. So you can, this shows it well. So you have this original cluster of proteins. Gets repackaged. And now these can be sent out to wherever they need to go after they get put into a vesicle. So this one happens to show it's going outside of the cell. That'll do its job. Alright, this one shows it, instead of in that typographic, shows a artist rendition of it. So once again, stuff in the nucleus is getting sent outside of the ribosomes, then gets sent to the rest of ER. where it gets packaged and sent to the Golgi, gets modified a little bit more in the Golgi, repackaged, and then sent to where it needs to go. And we're going to talk more specifically about this process right here when we get into the structure of the cell membrane. Alright, so when we look at the structure of the cell, you guys have a drawing you're supposed to be going through and making. You can see when we look at an animal cell, these are the structures that you typically will find inside of an animal cell. These are the things you're not going to typically find in an animal cell. You're not going to find chloroplasts. You're not going to find central vacuoles. Right? You're not going to find a cell wall. You're not going to find plasmodesmata. And we'll talk about those kinds of things when we get into the cell energetics unit and the photosynthesis. And we'll spend a little more time looking specifically at the plant cell. When you look at a plant cell, though, you're not going to find all the same things as you find in the animal cell. So you can see the general structures that are found there. And I believe you have this drawing in your textbook, and so you should be able to find this for yourself as well. But you're not going to find lysosomes inside of a plant cell. You're not going to find centrioles to be able to help guide the cell division. You're typically not going to find flagella. Typically those are only going to be found in animal-like cells. Alright? So I'm hoping you feel comfortable with these things. There's a lot of review from general biology, but it's also a little bit of an extension because we definitely went into a little bit more depth with that than we did in general bio. So now let's do the third tour. I know it seems like, oh golly, it's going to keep going on and on and on. This one's going to take a lot less time. Alright, so remember, we've already looked at the job of making proteins. We just got finished with that. When I come back tomorrow, we're going to talk about how cells make energy, at least give an overview, and we'll spend more time on the cell respiration piece. But now let's look at how is the cell designed to be able to make more cells for the purposes of growth, repair, and renewal. When we look at how new cells are getting made, you need to have some sort of structure inside the cell to be able to transport things to wherever they want to go. Back in general biology, we talked about having the cytoplasm and saying it was just this liquid layer where everything was sitting and everything just flows through the liquid. Well, you know what? We found out that no, that's not necessarily the case. We kept it simplified for you guys when you were 10th graders. But now let's look a little bit more specifically at it. There's actually kind of a skeletal structure system inside of the cell that is used for being able to transport things from one location to another within the actual cell. Right? It helps to maintain the shape of the cell. It then provides anchorage for organelles. Some of the videos we watched, you saw the little the proteins that were kind of walking on this thing that looked kind of like a road. We watched the intro to cells yesterday. Excuse me, it was on Tuesday or Monday. Yeah, whatever day we watched it. Anyways, you saw it kind of walking across that. That was an example of one of the structures inside the skeleton, inside of the cytoskeleton. So it provides anchorage for organelles. It also provides a method or kind of like a highway for things to travel throughout the cell. And I have a video. I was going to show it today, but I think I'm going to wait and I'm going to use that as our intro video on Thursday. That'll show that much better than the way I'm explaining it right now. Alright, some of these proteins that help to be able to make this anchorage thing, we call them, there's microfilaments, there's intermediate filaments, and there's microtubules. Alright, and this picture shows them in actuality. You notice once again, very small shot here. I have another picture later on in this that actually shows them in an artist rendition that helps to clarify exactly what these things are, I think a little bit better than what you see here. Alright, this helps with motility as well, because the cells need to be able to not only move around themselves at certain times, they also need to be able to efficiently move things throughout the inside of the cell. Because back in the 1940s, and back in the 1950s, all these interstates that we drive on now, they didn't exist. People had to take all these rural roads and all these little dirt tracks to be able to get to where they wanted to go. So we think about it now. We want to take a trip to Detroit and go check out a Tigers game because we want to see Fielder and Cabrera back to back. That's going to be flipping awesome. I can't wait for baseball season. If you didn't know it's a baseball fan, you just figured it out. Anyways, it took a lot longer than 2 hours to get to Detroit back then because of the fact that you had to take all these back roads. They built this interstate system so that we could more efficiently move from one location to another within the United States. Well, your cell has a very similar system. It has a series of highways that it can have things travel that will help get it from one location to another much more efficiently and much more quickly. So it doesn't have to just randomly send it out and hope it gets there as it's floating through all this fluid. There's things to help anchor it in place to help get it to exactly the location it needs to get to, like our roads do for us now. Alright, some of the other structures you're going to find cilia and flagella. We talked about those a little bit already. Cilia being little hair-like things that are on the outside of the cell to be able to, and has various things, but typically helps to be able to move the cell or to be able to move other particles across the surface of the cell. And they often help with being able to absorb things. And then flagella are typically things that are on animal cells that help them to be able to move. Best example of a flagella is the tail on a sperm cell. Alright, and then it helps with regulation as well. It helps to be able to control the activities that are going on. Once again, more so because it gives it a way for the proteins that help to control that. Gives them a way to be able to get from here to there a lot easier. So like I said, I think this picture helps to give more justice to the microfilaments, the intermediate filaments, and the microtubules. So here you can see the intermediate filaments. These are going to help connect the microfilaments and the microtubules together. And then, like I said, it provides kind of like the cross streets here. Then you have the microfilaments and the microtubules are kind of like the highways that are going to provide an area for things to be able to travel throughout the cell. So just recognize the cytoplasm, like we talked about before, is not just this fluid layer. There's other proteins that help to be able to guide things throughout the cell. There's also, when we're talking about cell growth and having the ability to divide, there's also a structure called the centrioles. These are going to help run cell division. You can hear it's 9 o'clock, by the way. You'll hear the dings. Anyways, with the cell division, there's these little structures called centrioles. And in some of our biology classes we talked about these, others we didn't. But hopefully this picture looks familiar, and I'm hoping even a couple of you can figure out that we're really looking at mitosis. And we're looking specifically at metaphase here, because it looks like all the, this is misrepresented. All the DNA is lined up in the middle here. Well, remember we talked about these little spindle fibers? Well, these spindle fibers are really nothing more than microtubules that are specialized to be able to help with cell division. Well, the areas in which these have grown out of, we call those centrioles. So you can look over here, you can see the centrioles, and you can see the centrioles are just a big cluster of a bunch of microtubules coming together. So in animal cells, you're going to find a pair of these centrioles that are nothing more than a grouping of organized microtubules, and then they start to connect to the DNA so that you can split the DNA evenly apart later on in anaphase during the cell cycle. In the specialized name we give these microtubules, when we're talking about them being involved in cell division, we call them spindle fibers. Once again, I hope that term is familiar to you. Alright, so these are the things that are going to help guide the whole process of mitosis. That's all I have on being able to use it for making new cells. So if you have any questions, I hope you've been recording them. We can talk about them on Thursday. I'll make sure I have a little bit of time set aside for us to be able to discuss those. And then we'll start getting into some of the structures that are involved in being able to provide energy for the cell. And then on Friday we'll start talking about, okay, let's get into the cell membrane and how the cell membrane is able to regulate the movement of things into and out of the cell. Alright, so I hope you have a good day. The last thing I'm going to have you do today, and this is depending on how much time there is, you should be getting a little worksheet that has a bunch of the different little structures that are found in various cells. I want you to make a Venn diagram. It's going to be a triple Venn diagram. One of the circles is going to be the plant cell. One of the circles is going to be animal cells. And the final circle is going to be bacteria cells. And what I want you to do is see if you can fit where we find those in each of those three types of cells. And appropriately fit it into whichever section of the Venn diagram is supposed to go into. Alright, if you have questions, like I said, we'll talk about those on Thursday. Hope you guys have a good day, and if those of you that were planning on coming in at lunch and working on test corrections, things of that sort, I am in the building. I'll still be there to help you guys out with that. Have a good day.